How are you all? I hope you are doing fabulous in this online lecture series. Well, today we will learn a lesson of standard 7th, which is 11th lesson from your textbook of subject science, which is a very interesting subject. That lesson's name is cell structure and microorganisms. So, in this lesson we will learn various types of subunits with the help of PPT, that is PowerPoint presentation. So, let's get started with that PowerPoint presentation.
As we see, letters together form words, words together form sentences, then sentences comes together to form paragraphs. After paragraphs, these paragraphs together forms chapters, and chapters are together form book. Similarly, this case we can see in the living organisms also, means organizational levels in living organisms. These are cells together forms tissues, tissues together forms organs, organs together comes and forms organ system, and these organ system together forms an organism, which may be human body or any living organisms like plant, animals include in this figure. You can see horse is given. So this is an example of organizational level in living organisms. So as we know that these cells form the basis of structure and function of all living organisms, it is only with the help of cells that living organisms carry out all their different life processes. So for learning those informations about cell, today we will learn the subtopic basic information of cell. In 1665, a scientist, Robert Hooke, whose picture is given on the screen, he observed a thin section of cork under a microscope which is given on the screen on right hand side. Then thereafter he saw a hexagonal compartment like those of a beehive. He named those compartments cells. This term is derived from the Latin word cella, which means small room. You can see clearly in the picture which is above the compound microscope, which shows a hexagonal compartment which he observed in his compound microscope. In 1838, the two scientists M.J. Schleiden and Theodor Schwann formulated a theory about cellular structure, which stated that all the living organisms are made up of cells, and the cell is the fundamental component of all living organisms. Then in 1885, as per your textbook, Rodolphe Virchow stated that all cells are formed from the pre-existing cells. Then measurement and observation of cells. In 1673, Anton van Leeuwenhoek assembled various lenses to construct a microscope. He was the first to observe live bacterial and protozoan cells under the microscope. As you can see in the picture given on the right hand side of this screen, compound microscope is given. Compound microscope have several parts like eyepiece, or it is called as ocular, as you can see in the figure. Then coarse focus knob, fine focus knob is there. Fine focus knob is for fine adjustment to clearly see that image. Base on which the microscope stands. Then illuminator is for focusing the light adjusting the light rays to get focused on that cells. Then stage is there, then objective lens, tube is there. These are all the parts of a compound microscope. Cells are extremely minute in size. We cannot see these cells with our naked eyes. Micrometers and nanometers are the units used for measuring their sizes. As we have seen lastly, compound microscope is used for observing these cells as they are very minute. An object on a glass slide magnifies these cells with several times by the lenses of this microscope. As micrometer and nanometer are the units used for measuring the sizes of cell, now we will see unit conversions. 1 centimeter is equal to 10 millimeter. 1 millimeter equals to 1000 micrometer. Micron is the unit here. And 1 micrometer or 1 micron equals to 1000 nanometers. Means you can imagine how minute these cells are. Here is the image of the activity to be performed to visualize the cells of onion under a compound microscope. For that, what you have to do is you have to take a piece or segment of onion and carefully separate the thin skin from its concave surface with the help of a forceps. As you can see in the figure, a layer is separated. Place the membrane on a glass slide and you have to put a drop of water on it. But while doing this, you have to take care that the membrane does not get folded while placing on the slide. Put a drop of a dilute solution of iodine or eosin over it to observe it, means to stain those cells. With the help of this eosin and iodine, the cells will be stained, means colored, and you will be able to visualize those cells. Then thereafter you have to observe these cells under objective of 10x. Here you don't forget to put a cover slip over the onion skin on the slide before placing it under the microscope. In this way, you will be able to observe the plant parts of various plants like leaves, bark, root tips, etc. So, in this lecture, we have seen organization in living organisms, means cells form tissue, tissue form organ, organ forms organ system, and organ system forms body. Thereafter, we have seen discovery of cell, which was given by Robert Hooke. Thereafter, we have seen cell theory, which was given by Sladen and Shawn and Virchow. Thereafter, measurements and observations of cells. How compound microscope is used to visualize these objects or cells and their measurements. This was all about this today's subunit. If you have any difficulties related to this subunit, you can ask in the comment section below this video. The last part is about homework. Your homework is you have to write what is cell and who first time observed the cell. Okay, thank you.